Next is the nucleic acid. The nucleic acid means what? That is the in every generation we have, that is each and every species of resemble, that is ancestor in many ways. These are the characteristics transmitted from one generation to the next generation. See, that means it has been observed that the nucleus of a living cell is responsible for the transmission of the inherent characters. So this is called heredity. The particles of nucleus of cells that is responsible for the heredity, and the chromosomes which are made up of the proteins and another type of the biomolecules is called nucleic acid. See, these are mainly two types. One is DNA, another one is RNA. The DNA is you are studied in biology, deoxyribonucleic acid, deoxyribonucleic acid, and the RNA is ribonucleic acid. So that nucleic acids are a long polymers of the nucleotide, so they are called polynucleotides. Usually they are called polynucleotides. Then the chemical position of the nucleic acid. Chemical position of nucleic acid. Here, the chemical position of the nucleic acids are, that is the complete hydrolysis of DNA or RNA yields a pentose sugar. That phosphoric pentose sugar and pentose sugar, not pentose sugar, pentose sugar. The pentose sugar, phosphoric acid, and the nitrogen containing the heterocyclic compounds, that is called base. Pentose sugar and phosphoric acid, and the nitrogen containing heterocyclic compounds are called bases. See, in a DNA molecule, the sugar moiety, that is beta D2 deoxyribose, whereas in RNA molecule, beta D ribose. This is a RNA molecule and this is a DNA molecule. See the difference of these two. Why this is beta D ribose, why this is beta D2 deoxyribose, means here the OH, look at this, the carbon number we can give here. This is first carbon, second carbon, third carbon, fourth and fifth. So this having a pentose sugar, and here also 1, 2, 3, 4 and 5. Look at here, beta D ribose and beta 2. So the position of 2 indicates what? The 2 hydrogen are present here, but here in this hydrogen and hydroxide ions are present. So that this different. This is beta. Others molecules are same. That is in RNA and the DNA difference only in the OH group instead of the hydrogen group in the second carbon atom. So that is why this is beta D2 deoxyribose. This is beta D ribose. So DNA contains the 4 bases like adenine, guanine, cytosine, thymine. So these are the main, and uracil. That RNA also contains 4 bases. That's why the first 3 bases are the same. That's mean adenine, guanine, cytosine. These are the fourth one, that is the uracil, and the thymine is different. For see the adenine, the structure of adenine, N double bond with CH. NH, CnH2, NCH, and N. So this is the structure of adenine. Simply A. So here the DNA contains DNA and RNA contains the bases. The DNA contains the bases adenine, guanine, cytosine, and the thymine. So these are the bases of the DNA. The RNA contains also three, that is adenine, guanine, and cytosine. The fourth one, that is only differ. This is called the uracil. Uracil is the RNA bases. So these are the base structure of the DNA and the RNA molecule. Then the structure of the nucleic acid. The structure of the nucleic acid. Nucleic acid, that the base to one dash position, simply sugar, that is the sugar carbons are numbered like this: 1 dash, 2 dash, 3 dash, 4 dash, like this. See, in order to distinguish these form of the bases, like when the nucleoside linked to the phosphoric acid, the 5 dash position, this is called sugar moiety. I will write the structure here. The structure of nucleoside and the nucleotide. Here the bases contain. This is the base. H, OH, H, OH, H, CH2, OH. See, this is a nucleoside, structure of nucleoside, and this is nucleotide. Here the base, that is 1 dash, attached to the base position, that is it is called a sugar moiety. That's the sugar carbons are numbered like this: 1 dash, 2 dash, 3 dash, 4 dash, 5 dash. Here also the same way follow. The nucleosides are that see, the nucleotides are linked together, that is the phosphoric acid in the 5th position. So this is the phosphoric acid, the 5th position. This is a sugar moiety. So you will be getting the nucleotide. Here just does not attach any sugar moiety, but here the phosphoric acid of that 5 dash position, it is called sugar moiety. The nucleotides are joined together by phosphodiester link also. See, here the nucleotides, the nucleotides are joined together. The linkage between the phosphodiester linkage. This is the phosphodiester linkage. See, the 5 dash position in this, the 5 dash position and the 3 dash position, that is the carbon atom. The 5 dash and the 3 dash carbon atoms are pentose sugar. This is a pentose sugar. So the formation of the typical dinucleotide. This is dinucleotide. Okay, this is the formation of the dinucleotide structure. That is the simplified structure. We can write like this: sugar, base, phosphate, sugar, base, phosphate, this goes n numbers, then sugar, base, like this. So this is the simplified version of this dinucleotide. The sequence of the nucleotides in this chain of a nucleic acid are called a primary structure. So the nucleic acid have the secondary structure also. That is the double stranded helix structure. That is, two nucleic acid chains are wound about each other to held together by the hydrogen bond between the pairs of the bases. So the two strands are the complementary to each other because the hydrogen bonds are formed between the specific pairs of the bases. So the adenines forms the hydrogen bond with thymine, whereas the cytosine forms the hydrogen bond with guanine. So the secondary structure of the RNA, that is helices are present, which are the only single stranded. That sometimes they fold back themselves to the double helix structure. That is RNA of the three types that is perform a different function. That is the name that's called messenger. Messenger of RNA, that is simply indicate mRNA, and ribosomal RNA, that is simply rRNA, and the transfer of RNA simply tRNA. This is ribosomal RNA, transfer RNA, and this is the messenger RNA. Okay, and the double stranded helix structure of DNA also there. That is the structure like this. You can see in your NCRT like this. Okay, that is GCAT, like this. GACT indicates that is guanine, cytosine, thymine, adenine. These are the name. Then the biological function of the nucleic acid. Before that, the DNA fingerprinting. The DNA fingerprinting, every individual person, that is the unique, they have a unique fingerprint. These occur at the tips of the finger and have been used in the identification of for a long time, but these can be altered by surgery. You can alter by the surgery. So the sequence of bases on a DNA is also unique for a person, and the information regarding this is called a DNA fingerprint. So every cell and cannot be altered by any known treatment. Okay, so DNA fingerprint used are the forensics laboratory. Forensics laboratory for identification of criminals, determine the parentage of the individual, and identify the dead bodies of any accident by comparing the DNA of parents or the children, whatever it is. Then they identify the racial group to rewrite the biological evaluation. So these are the useful for the DNA fingerprint. Then the biological function of nucleic acid. See, the DNA is a chemical basis of heredity and may be regarded as the reserve of the genetic information. So here the DNA is the exclusive responsible for the maintaining of the identity of the different species of organisms over the millions of years. So that is the DNA molecule is capable of self-duplication during the division, and the identical DNA strands and the transfer to the daughter cell. So that the important function of the nucleic acid is the protein synthesis in the cell, but actually proteins are not synthesized by various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in the DNA. So this is the function of the biological function of the nucleic acid. Okay, student, thank you.